Hey Fiona, hope you're doing well. We're going to have to do homebound this week, just kind of virtually, but without actually talking. So please email me with any questions that you have. But last week, we got cut off a little early, but you should have finished looking through the PowerPoint. We looked through different organelles, and it was pretty straightforward on the organelles and what they did, really. So we looked at lysosomes, Golgi bodies, endoplasmic reticulum, that sort of stuff. So it was just basically, here it is, and here's what it does. There was a YouTube video here that you could have clicked on. You should have clicked on and watched that video. And then I asked you to work on the Section 1 review questions on Google Classroom under cells. So today we're going to move on, get a little bit new notes, and then I'll tell you what I'd like you to do. Okay, so we're still on our cells chapter, and today we're going to talk about magnifying cells and how people look at cells. Now we know that cells are the smallest units that are considered living. Okay, so the number of things you can't see in your environment, so the number of things you can't see around you in your daily life is way, way, way bigger than the things that you can see. For most cells and a lot of things in your environment, you need a microscope to be able to see. So this website here we're going to look at, it's a really cool website. It shows us, and I have it up here, it's the scale of the universe, and it's just scaleoftheuniverse.com. You can go there and play around if you'd like. But it shows us here that if this was the size of a human, you could see many things that you would recognize. A basketball. So you could think of this human holding that basketball. That's in scale, right? That's the correct scale. You could imagine that person holding a basketball. You could see a ruler here, a teapot, okay? Now, if we scroll in, and we'll scroll out too because that's fun, but when we're talking about cells, we're talking about things that are smaller than a human. So if we were to scroll in, you could see a hummingbird in relation to a human, in relation to a basketball. There's the largest hailstone that was ever measured that we know of. And then we go down in here, we see a shrew, a chicken egg, a matchstick, things that you could still see, things that are still very common to you. A penny. Okay, here's what a square inch would be in that measurement. A microwave wavelength would actually be pretty big here. A coffee bean, the largest bacteria we know of, an ant, a sunflower seed. Still things, pencil lead, okay, there's that 0.7 that we typically use. A grain of rice. And then as we get down here, we start to see a grain of sand. You could still see a grain of sand, right? You could still see the width of a human hair, but it starts to get very small on us very quick. Here we have a grain of salt, a dust mite, an amoeba. Okay, so here we go into things. An ovum, which is a human egg. This is really the smallest object visible to the naked eye. So you would be able to see a human egg cell. A mist droplet, a paramecium. So even paramecium dust mites, they're observed, although tiny and we don't often notice them, we could technically view them without a microscope. Once we get smaller than this, you're going to need a microscope. So as we get down here, we see a skin cell, okay, width of a silk fiber. That's why silk is so soft, because the fibers are so fine. I get down in here, thau-twip, I don't know what those are. But we have white blood cell, red blood cell, okay, a cell nucleus. That's something that we went over last week in the slides. So once you saw that, a chloroplast, another thing we went over last week. Chromosomes are found within that nucleus. Viruses we'll be going over later. Mitochondria, another one of those organelles. So you go down here and you can see how small viruses are and DNA. Okay, get down in here. Phospholipid bilayer, that's a cell membrane. Okay, a glucose molecule, that's a molecule of sugar. You've got your phospholipids, which make up your cell membranes. Then you start getting down in here to atoms, a hydrogen atom, okay, a helium atom. They're very tiny. So you can go in there even more, and we get into these wavelengths and nuclei of different atoms with the protons and the electrons, okay? And we get, so lengths any shorter than this are not confirmed because we start to get really, really small. So we have quarks, stuff that you won't learn about for a very long time, if ever. But then as we scroll out here, what's also fun is if we see, go back to the human. Okay. It's also fun to scroll out and see things that are bigger than us, because I feel like we often feel like we're so big, and then we see all these other things in the world that are larger than us, and it's really a weird concept. But it's also strange to think of how small we are, how big we are. So that's why I like this website. But if we scroll out, you can see the dodo bird that's extinct. That was a big bird compared to humans. You see a beach ball, a sunflower, things you know of. And then a giraffe, if you've ever been to the zoo, or an elephant, you've seen those things before. But then other things like here's a human versus a T-Rex. I know if I saw that coming at me, I would probably pee. A Japanese spider crab that actually exists, a cactus. Okay, here's, you can still see a human versus a blue whale, a human versus the average house, an oak tree. Here's another type of dinosaur. Here's a human versus the Statue of Liberty. So that shows you how big the Statue of Liberty actually is. Here's a football field compared to a human. We know, if you're, watching, if you've ever seen football on TV or whatever, you could see how small from those aerial views, how small all the people are. But here's how big the Titanic was. So the Titanic was bigger than the Statue of Liberty, which blows my mind. We've got the Hoover Dam, the Eiffel Tower. Okay. So I always thought the Eiffel Tower and the Statue of Liberty were around the same size, but that's definitely not true. The Eiffel Tower is way larger than that. Hold on one second. My phone's ringing. All right. So I'm back now, but that is the site, and you can go all the way out here. You can go to, different comets, states, stars. Okay. California. So also on a map, I always think that California looks so much bigger than Italy, but in reality, they're about the same size. So you can go out here to the planets and whatnot. So that's just something for you to play around with if you want to go to that website. Okay. But just tells you a lot. But we're going to focus on, like I said, the cells. And in order to view cells, because they're so small, we often need microscopes, right? So our early microscopes, really early on, 1600s, they were very small. So this is kind of what they looked like, and you held it up to your eye. So this guy, Anton von Leeuwenhoek, in the 1600s, he used a little glass bead as his lens, and it was able to magnify things up to 270 times. So things look 270 times bigger than they actually were. But those images weren't always very clear. Okay, so you could see them, but it's blurry. And he saw living things in moving around in pond water. So that was a big discovery back in that time. Modern microscopes, depending on how many lenses a microscope has, is whether a microscope is classified as a simple microscope or a compound microscope. A simple microscope is more like a magnifying glass. It has one lens, and the lens makes an enlarged image of an object and directs the light towards your eye, okay? And when we talk about an enlargement of an image, we call that enlargement magnification. So we'll say that the image has been magnified. It's bigger. A compound microscope uses two sets of lenses. There's two lenses in the eyepiece here, as I'm circling. And then you'll have lenses in your objective lens down here. That's typically two lenses down here as well. And these move so you can view something under low, medium, or high power. So this is the type of microscope that when you get up into high school with me, we will be using these microscopes here. Okay. So when we talk about our microscopes, we talk about total magnification. So what we do for that is we take your eyepiece lenses and we multiply them times your objective lenses. So let's say our eyepiece magnified 10 times and our objective lens magnified 40 times. Well, 10 times times 40 times is a total magnification of 400 times. So whatever we are looking at underneath that microscope would appear 400 times larger than it actually is. Okay. Electron microscopes, they're pretty fancy-dancy. If you look at this picture here, the microscope I just showed you, that compound light microscope, could fit on your desk. And an electron microscope is typically the size of your desk, if not larger. So that's what you see here. And they don't use lenses or light. They use beams of electrons, and they use magnets. And then the images, they like detect the surface of an image, or they'll go through like a thin slice of an image. And then that specimen, the image of that specimen, is produced electronically with a computer. So several kinds of those, and we'll study those more in high school. But there's your scanning electron microscope, and that it scans the surface through, let's see, that's through so it's a spray that will actually go on the specimen, and it detects the surface. The transmission electron microscope, the specimens for that need to be pretty thinly sliced. And then the scanning tunneling electron microscope detects electron clouds of substances using a sensor needle, and it goes over, like I said, the surface. Okay. So as time has gone on, the advancement of microscopes has allowed scientists to learn about cells, to study cells. And in 1665, a guy named Robert Hooke named cells cells. And he was a monk, and when he was looking at he was basically looking at dead plant cells under the microscope, and he saw all these little square boxes. And he said, these remind me of little rooms. And the rooms where he lived, monks lived in monasteries, those rooms are called cells. So he named, because they reminded him of rooms, he named cells cells. And many scientists worked with cells, and 3 of which have their work summarized in what we now know as the cell theory. And those scientists were Schleiden, Schwann, and Virchow. And the cell theory basically says this. There's 3 parts. Living things are made of cells. Okay. Next, cells are the basic unit of structure and function in living things, meaning they are the smallest things that exist in living organisms that work. Okay. And can be considered alive. And then finally, cells come from other cells. Cells can only produce cells. You're not going to get cells from organisms that really aren't living. Okay. So those 3 things come up with the cell theory. And I have a video on microscope history that I'll be posting for you to also watch. And that's about 10 minutes. We're going to get into our next video for you to watch over the next week on viruses in a hot minute. I'll be with you again shortly. Thanks, Fiona.